Hello everyone, welcome back to another video lecture on sexual reproduction in flowering plants. In this video, we're going to be study about the embryo, the development of the embryo and the dicot and monocot embryo. We are going to be studying this video lecture. And in this video lecture, in the last previous classes, we are already started the post fertilization events, and in that post fertilization events we can find the series of steps. Okay, it includes the development of the endosperm. This is the first one. Second, embryo development. Third one, maturation of ovule into seed and ovary into fruit. Okay, this is the criteria of post fertilization events. Okay, these are all going to be happen in post fertilization events. Already we studied about pre fertilization events and fertilization events, and now these are the post fertilization events. In this we already studied about the endosperm, what is endosperm and the types of endosperm. Okay, usually the polar nuclei, the middle there is, in the central cell there is a presence of polar nuclei. They are going to be fused with one gamete and they will form endosperm. Okay, function, it is mainly nourish the developing embryo. Okay, mainly nourish the developing embryo. That is the function of endosperm. And the endosperm can vary. Okay, there are mainly two types of endosperms we can see here. What are those? The first one, that is pre-nuclear endosperm and then cellular endosperm. Pre-nuclear endosperm means there is no division of the cell, or there is no cytokinesis. Okay, the nuclei is going to be divided, but there is no formation of the cell wall, so it will not become a cell. Okay, such such endosperm we call it as a pre-nuclear endosperm. We can take the example for this coconut. Coconut is the example for pre-nuclear endosperm. And coming to the cellular endosperm, what will happen? Nuclei are going to be divided, and then there is a development of the cell wall. There is a development of the cell wall. So there is a cellular endosperm. This we call it as a cellular endosperm. Here also you can take the example coconut. Okay, you can see the white portion inside the coconut. That white portion is cellular endosperm, and the liquid portion is nothing but pre-nuclear endosperm. This already we studied. Here they given two example, coconut and castor. Okay, and then in case of peas and beans, you can see the and here one thing they given another one concept, that is in some cases what happen, the endosperm is consumed completely. Say for example, you can take pea and beans and groundnut, you can take those example. The endosperm is going to be consumed completely, and in some cases what happens, it is going to be retained. Okay, it is going to be retained in some mature seeds, such as example you can take coconut and castor. Okay, two example they given. Okay, the endosperm is going to be consumed completely. Such and endosperm we call it as a non-endospermic seeds. Okay, in some cases what happens, endosperm retained. Okay, after the complete formation of the seed. Okay, germination of that, during the germination of seed, the remaining endosperm is going to be utilized. Okay, the such seeds we call it as a endospermic seeds. Okay, this is the endosperm. These are all the points regarding endosperm. And moving to the next concept, that is embryo. Embryo we are going to be studying both in case of dicot and in case of monocot. In case of dicot and in case of monocot, we are going to be study about the embryo, how the development of the embryo is going to be takes place. In your NCERT textbook, the development they are not given. Okay, in your NCERT textbook there is no explanation about the development of the embryo. They only given the dicot embryo and monocot embryo, their parts. Okay, that much only they given, but they given the diagram about dicot embryo. That I will tell you in a little, in a small way I will tell you that. And first here about the embryo. First what will happen, in the micropillar end there is a formation, and here I will show the diagram first. Okay, here, okay, this is the, sorry, this is the diagram. This is the fertilized embryo sac. You can see here the fertilized embryo sac. there is a presence of primary endosperm, and then here the primary endosperm nucleus. This is the primary endosperm cell, and this is the primary endosperm nucleus, which is triploid. Okay, this is going to be become a that is endosperm. It is going to become a endosperm, and here you can see the zygote. Not here, sorry. Here you can see the zygote. Okay, this is the micropillar and this is the chelagel end. Here you can see the antipodal cells, here you can see the synergid cells, and then egg cell. Here exactly the zygote is going to be formed. Okay, double fertilization occurs here, and then there is a one zygote and one endosperm is going to be formed. And here the zygote formed in the region of micropillar end. Okay, in the region of micropillar end, there is a development of the zygote. Okay, there is a development of the zygote we can see here in the micropillar end, and this is the chelagel end. Just remember chelagel end and then micropillar end. And here during the fertilization is going to be happen here with the help of synergid cells. There is a fertilization occurs with the egg cell, and before the development of the embryo, okay, before the development of the embryo, there is the development of endosperm, because this endosperm will give the nourishment, because zygote need much energy. Okay, it's a complicated process. The cells has to divide here continuously. For that purpose it need energy, and that energy is mainly produced by endosperm. So as early as possible there is a development of the endosperm. Okay, up to a certain amount of endosperm is going to be formed. When the certain amount of endosperm is going to be formed, at that time there is a development of the embryo begins. Okay, this is the concept. First embryo is not going to be developed. First endosperm is going to be developed, and later on there is a development of the embryo. Okay, because embryo, this endosperm will give the nourishment to the developing embryo. That is the thing. Okay, and here what happens, this is the complete explanation about the development of the embryo in dicot, not in monocot, in dicot. This is the zygote you can see here, and this zygote is going to be divided unequally it is going to be divide here, unequally it is going to be divide here. And one is apical cell, another one is basal cell. Okay, one is apical cell, another one is basal cell. Here this is the apical cell, this is the basal cell. This is the basal cell and this is the apical cell or terminal cell we can say. And it is going to be divided unequally. Okay, when the energy is going to be produced from the endosperm, the zygote will start division, but the division is unequal. In the beginning the division is unequal. So one is basal cell, another one is apical cell. You can see here clearly. And later on what will happen? The basal cells start to divide, and there is a formation of suspensor cells. Here these are all the suspensor cells. These are all the suspensor cells. And the last cell of the suspensor cell, here you can see, this is another cell. Okay, large cell is present here. Okay, like it is called as what you say, this we call it as a this we call it as a last cell, which is towards the micropillar end. Sorry, it is towards the chelagel end. Not this one, this one chelagel end. This large vacuolar cell we call it as a hostorial cell. Okay, it is called as hostorial cell. This cell we call it as a hostorial cell, which is towards the micropillar end, the last cell of the suspensor cell, hostorial cell we call it as. And here you can see the structure, that is an embryo. Okay, embryo usually here zygote, embryo development here you can see here, this is the embryo development. And here you can see the different structures of the embryo. Okay, different structures of the embryo they given in your NCERT. One is a globular structure. Here they given the globular structure. This structure we call it as a globular structure. And next to that, heart shaped structure we can see here. Heart shaped structure here, it looks like a heart, it looks like a heart. So this we call it as a heart shaped structure. And whatever the structures you are seeing here, these are cotyledons. These are nothing but cotyledons. Okay. And here the last structure, that is mature embryo. This is the matured embryo. You can see here the suspensor cells, here the cotyledons you can see here, and then the custodial cell you can see here. Okay, this is the structure what we are seeing here. And this is the structure, and this much only given in your end, not this much also. They are not explained about the embryo in your NCERT development of embryo. They given only the direct the structure of dicot embryo and the structure of monocot embryo. And if you move to the next structure, I think so it somewhat it is understandable, I think so. And if you move to the next slide, that is coming to the um, dicot embryo. Okay, this is the dicot embryo what we are seeing here. This is a structure. Please remember, this is also important structure. They can ask the diagram for 2 marks. Draw neat level diagram of dicot embryo, or draw neat level diagram of monocot embryo. They can ask for 2 or 3 marks. So important, please remember here. And here they given the structure. If you observe the embryo, okay, whatever the embryo you observed over there, it was like this. It was like this, and heart shaped here like this is the structure of embryo what we studied here last. And if you observe, if you bend that structure slightly towards this, you can see the structure like this. Okay, if you bend that structure slightly, here these two cotyledons you can see here, these two are the cotyledons, and this is the your suspensor cells and everything you can see here. Okay, and if you observe the structure of dicot embryo, you can see here the embryonal axis and two cotyledons. You can see embryonal axis. I will draw here. This is the embryonal axis. You can take, this is the complete embryonal axis. Okay, these two are the cotyledons. They already labeled here, cotyledons. And where exactly the cotyledons are present? Just the structures above the cotyledons. You can take the axis of cotyledons like this. This is the embryonal axis and this is the cotyledonal axis. Above the cotyledonal axis, okay, the structures above the cotyledonal axis, that structure we call it as a epicotyle. It is not labeled here, but they mentioned in explanation. This is epicotyle, just mark it. This is the epicotyle, which can terminate as a shoot tip or flumule. Already this you studied in first year only, morphology of flowering plants. Okay, flumule and radicle already you studied. So if you observe here, the above the embryonal axis or above the just cotyledon, okay, the above the axis of the cotyledon, that we call it as a epicotyle. In that epicotyle you can see the ending of the epicotyle, or this epicotyle is going to be end as a shoot tip or flumule. We call it as a flumule, which can give rise to shoot. Okay, which can give rise to shoot. This structure we call it as a epicotyle. And below the embryonal axis, or just below the cotyledons, okay, it means the lower end of the cotyledon region, there is a presence of hypocotyle. Okay, which is present above, that is epicotyle, and which is present below the level of the cotyledon or the lower level of the cotyledons, that we call it as a hypocotyle. And this hypocotyle is going to be end with a radical and a root cap. Okay, radical and root cap. Here you can see the radical, and this radical is mainly covered by root cap. This radical is mainly covered by root cap. This is the structure of dicot embryo. This is the structure of dicot embryo. This is the embryonal axis. Sorry, this is the embryonal axis, and this is the cotyledonal axis. You can take just once I will explain here. This is the embryonal axis, Okay, this is the cotyledonal axis you can take, and this embryonal axis, the above the level of the cotyledons or the upper level of the cotyledon structure, we call it as a epicotyle, and this epicotyle is going to be end with flumule, and in the lower level we can see here hypocotyle. This structure we call it as a hypocotyle, and this hypocotyle is going to be end with radical and root cap. Radical and root cap. This is the structure of dicot embryo. Okay, this is the structure of dicot embryo. Very simple. Wherever the parts are there, you can observe that. And these two are the cotyledons. These two are the cotyledons. Okay, these two are complete structure. These two are, we call it as a cotyledons, which is dicotyledon. There is a presence of two cotyledons here. Two cotyledons are present here, so it is a dicot. Okay, I think so you understood the structure. Only what they given, only the parts they given, the parts of the uh, embryo, dicot embryo they given. Okay, this is the structure. And if you observe the points regarding dicot embryo, here they given the points. Okay, here the embryonal axis is present here and two cotyledons are present here, and above the level of the embryonal axis we call it as a epicotyle, and it contains a flumule or the shoot tip. The portion below the axis, embryonal axis, is called as hypocotyle, and it ends with the radical and the root tip. Okay, root tip is covered with the root cap. These are the points regarding dicot embryo. Very simple points are there. Please remember all those. Okay, this is the dicot embryo. And if you move to the next one, that is monocot embryo. Okay, if you move to okay uh, if you move to the next monocot embryo, you can see the structure of monocot embryo. Okay, you can see the structure of monocot embryo, and these are all the points about the monocot embryo. Here you can see first the structure. Okay, this is the structure what you are observing here, that is monocot embryo, or grass you can take. You can take grass. Okay, monocot embryo is also same, and the grass embryo is also same. This is the grass embryo what they given, and they can ask the question, draw neat label diagram of uh, embryo of grass, or sometimes they will ask the question, draw neat label diagram of monocot embryo. Okay, but in your NCRT they given the diagram, this diagram only, they given it as a grass embryo. Grass embryo means it is a monocot embryo. Okay, please remember that. And here if you see the structure of monocot embryo, here also you can divide the embryonal axis and cotyledon. Here it is a monocot, so only one cotyledon is present here. Only one cotyledon is present here. If you take the embryonal axis, this is the embryonal axis and this is the cotyledonal axis you can take. If you go to the embryonal axis, this we call it as the embryonal axis. And the cotyledons in monocot, we call it as a scutellum. Please remember, define scutellum. They can ask the question like that. Okay, scutellum is nothing but a cotyledon in case of monocots. Okay, it is a cotyledon in case of monocots. Single cotyledon present here. Single cotyledon present here. Here also you can see the epicotyle and hypocotyle. The region or the part which is present above the embryonal axis or higher level of the embryonal axis or above the scutellum level, here the scutellum is attached, and above this level we call it as a epicotyle. Same here, we call it as a epicotyle, and below this we call it as a hypocotyle. Please remember, hypocotyle, epicotyle and hypocotyle, these two regions. And here in the epicotyle region, here you can find the scutellum. A single scutellum you can find here, or single cotyledon we can find here. And in the another side, okay, only one side, it is present in a one side, this scutellum present in only one side, and in another side there is a presence of epiblast, which is nothing but another cotyledon, but this cotyledon is not developed here. It is not developed because it is a monocot, so only one cotyledon should be there. For that, in one side there is a scutellum present, and on the another side there is a presence of a small projection. You can see here, this is the epiblast, and this epiblast is nothing but cotyledon. It is not developed well. Okay, it is not developed well, so it is a one cotyledon, you can see. And if you observe the next one, in the epicotyle region, there is a presence of shoot affix. There is a presence of shoot affix, or we call it as a flumule. We call it as a flumule. Okay, writing is not that much good, just understand about that. Flumule or shoot affix, or we can say it as a flumule. Okay, and this flumule is covered by a hollow covering, that is coleoptile. This shoot apex is mainly covered by a hollow membrane. Here you can see this hollow membrane. This is coleoptile. We call it as a coleoptile. Please remember. And these coverings and everything not present in dicot. These are present in monocot. What is the function? It mainly covers the flumule. Okay, it can help to penetrate through the soil. Okay, mainly give the protection to the flumule or the shoot apex. You can take the function of the coleoptile is protection. You can take the protection to what? Protection to shoot, protection to developing shoot, because it has to pass the soil layer. So for that they need a membrane, a strong membrane. Okay, strong membrane they need, and that is coleoptile. Okay, and this coleoptile is mainly protects the flumule. Okay, this is the thing. And here you can find the scutellum, already we studied. And next to that, if you come to the lower region, okay, lower region of the embryonal axis, this is the hypocotyle. This is the hypocotyle. And in this hypocotyle, you can see the radicle. And the lower, already we know that the lower portion is gives rise to root system, and the upper region gives rise to shoot system. Okay, upper region we call it as the epicotyle, and the lower region we call it as the hypocotyle. And the lower region, there is a shoot root system present here. You can see the radicle here. In the hypocotyle region, you can find the radicle here, and that radicle is mainly covered by root cap. Then it is mainly covered by root cap. Here you can see this is the root cap, and this root cap is surrounded by a undifferentiated membrane, that is coleorhiza. The undifferentiated membrane, that we call it as a coleorhiza. Please remember, the undifferentiated region which is the undifferentiated membrane which is covered the root cap. Okay, that we call it as a coleorhiza. And here coleoptile, covering of the flumule. Here you can see the coleoptile, that is upper region. In the upper region, coleoptile, that is covering of the shoot tip apex or flumule. And here you can see the, in the basal region, you can see the coleorhiza. You can see here the coleorhiza, and this coleorhiza is covered the root cap or radicle. Both the function is same. Coleoptile also protecting the shoot tip, and here the coleorhiza is mainly protecting the root tip or root cap. It is going to be protect. Two layers we can find here. Okay, two layers we can find here in case of monocot embryo. Okay, this is the explanation about monocot embryo. I think so you understood about the monocot embryo. Okay, once I will repeat the monocot embryo. There is a presence of embryonal axis. There is a presence of embryonal axis. Okay, you can see the cotyledonal axis here. Above the embryonal axis or in the upper region, there is a presence of epicotyle, and in the epicotyle region it contains shoot apex, which we call it as a flumule, and this flumule is covered by coleoptile. Okay, a hollow layer we can see here, that is coleoptile. And one side of the embryonal axis, above the embryonal axis, on one side there is a presence of a single cotyledon, that we call it as a scutellum. And coming to the lower portion or in the basal portion, or along with that, here you can find the epiblast. Okay, this is the another cotyledon present in another side. Okay, next side, opposite side, but it is not developed here, well developed, so we will not consider it as a cotyledon. And in the basal region there is the hypocotyle, and the hypocotyle mainly contains radicle, it contains root cap, it contains another undifferentiated layer, that is coelurhiza. Okay, there is the presence of coelurhiza, which is a covering of root cap or radicle. This is the structure of monocot embryo. I think all of you understood about this diagram. Okay, and previously about the dicot embryo. This is the structure of dicot embryo, you can see. And if you go through the points about the monocot embryo, here the points they given. Monocot embryo, there is only one cotyledon. Grass they taken the example, and one cotyledon we call it as a scutellum, and it is situated in one side of the embryonal axis. The lower end of the embryonal axis has a radicle, a root cap enclosed in a coelurhiza. Okay, undifferentiated membrane, coelurhiza. The epicotyl lies above the level of the scutellum. Where the scutellum is attached, above that level we can find the epicotyl. And here the shoot affects and leaf primordia enclosed here. And some another point here, some primordial leaf primordials are present here. Primordial means those parts gives rise to the leaves. Okay, such primordials are primordia are enclosed here in the hypocotyl region, in the epicotyl region. Okay, and this structure, a hollow structure is enclosed here, that we call it as a coleoptile. This is about the monocot embryo. Okay, what we study, student, here the monocot embryo and dicot embryo and the process of formation of embryo. Somewhat that process of formation of embryo is somewhat a lengthy process. Okay, that not given in your NCERT, but they given the diagram, and just remember the unequal differentiation, and along with that undifferentiated division, and then the suspensor cell, and then you can see the globular embryo and heart shaped embryo and matured embryo. Okay, so many differentiation in various types of differentiations can occur here. So this is somewhat complicated one, or it is easy if you understood the concept, and very easy to go through that anyway, but it is not given in your NCERT, so that I am not discussing here. Okay, this is about the embryo development. Okay, monocot development they are not given, but dicot embryo diagram they given here. This is the diagram. Okay, okay students, this is about the monocot and dicot embryo, and in the next class I will start with next concept. Okay, that is seed, the types of seed, structure of seed, that we will discuss in the next class. I hope all of you understood about this video, and if you have any doubt or any complex any difficulties, you can ask through WhatsApp, or you can ask the queries through WhatsApp, and go through the video and repeated times, and read NCERT. Okay, very simple concept, you can understand very easily. Okay, thank you, we will meet in the next video. Thank you.